Okay, hi guys. Can you hear me? Okay, so this summer I worked in a medical lab as an intern in Yale's Discover to Cure program for 6 weeks. And I didn't really know what I was going to be doing, and nobody else really knew either, so there were a lot of different perceptions that developed of what I did this summer. So, basically, it's my friends thought I did, just kind of like some weird, crazy science stuff. My parents thought that I spent most of my time doing normal things with my roommates, like building forts. Shout out to Allie Elkman right there. And Miss Monty thought that I'd be doing kind of intern things such as getting coffee and maybe cleaning a few test tubes if things got really exciting. But my lab workers thought that I mostly just broke their equipment. But it's okay because the other intern broke more than I did. The program was called Discover to Cure, so it's kind of that's what I thought I was doing. But this is what I actually did. So for those of you who don't know me, I'm Kayla, and I'm a senior, and this is what I studied this summer. So this summer, I focused on the brain component as the cross-link between energy and reproductive systems. Energy homeostasis acts like a scale, balancing consumption with expenditure. But how does this energy scale affect reproduction? Without enough fat storages, a woman can't survive if also supporting a pregnancy. So low energy storages will signal to the brain to pause the reproductive cycle. For example, anorexic women with too little body fat often don't menstruate. This energy scale also affects puberty. Often gymnasts, mostly who overexercise and lack body fat, undergo puberty much later than average. This happens because this energy imbalance signals the brain to stop growth and delay the onset of puberty. Too much body fat has the opposite effect, like this little chubby guy. So, for example, the rising trend in obesity directly correlates with the growing number of American children entering puberty younger than average. Premature puberty is partly caused by too much body fat, signaling that the body has energy to grow and can bear pregnancy, thus initiating puberty at a younger age. There are several energy balance signals that act on many different neurons in the brain. One of these neurons is the neuropeptide Y neuron. Neuropeptide Y, or NPY, is the chemical that I studied this summer. It exists naturally in the brain, and it helps control the food intake part of the energy scale. It's the most potent substance that will make you eat. So for example, when injected in mice, immediately afterwards the mice start eating, even if they weren't hungry before. NPY concentration increases during fasting, but once the body consumes enough food, proteins such as leptin and insulin will decrease NPY concentration. Previous research also revealed that NPY helps regulate the reproductive hormones and the reproductive system by inhibiting reproductive brain cells. It's actually an important control mechanism for the timing of puberty, acting as a brake on it. And it also helps regulate fertility. Now let's focus on the reproductive system. Dr. Albrich's lab, where I worked, studied neuroendocrinology, the basis of which is the hypothalamus-pituitary-gonadal axis, referring to the hormonal system that connects the brain to reproduction. The hypothalamus contains these cells in the red brackets, and it's a control center for homeostasis. So the kisspeptin cells, which I'll talk more about in a moment, excite these cells called GnRH neurons, causing them to secrete the hormone that they contain, gonadotropin-releasing hormone, or GnRH. GnRH then stimulates the pituitary gland to release its hormones known as LH and FSH. LH and FSH then excite the gonads, or the ovaries and testes, to secrete sex steroids, which then complete negative feedback loops with the pituitary gland and the hypothalamus. Negative feedback loops are important for maintaining appropriate hormone levels for proper functioning in development, puberty, and fertility. So now let's focus on kisspeptin neurons, as they are a very important part of the reproductive system. Kisspeptin neurons were just discovered very recently, in 2003 actually, and researchers found that they act upstream of GnRH neurons. Kisspeptin neurons are located in two areas of the brain, and we focused on those in the arcuate nucleus of the hypothalamus, because both sexes have kisspeptin cells there. Kisspeptin neurons release kisspeptin, which basically is a protein that causes GnRH neurons to fire and secrete GnRH. Back to MPY. What are the reasons we have to study MPY? So, although there are several pathways to regulate the reproductive system, we believe that the MPY pathway plays a major role, especially in this energy reproduction crosslink. But we don't know where and how MPY inhibits the reproductive system. We wanted to learn which specific cells MPY acts on. This could be kisspeptin cells, GnRH cells, or even both. Research also proved that MPY acts on 5 known receptors, but we wanted to learn through which specific receptors does MPY inhibit the reproductive system. So how do we go about studying MPY and its effects? First, we'd extract a brain from a mouse and cut it into slices 300 micrometers thick, which is really, really thin, about like half the size of a penny, I guess. This is the apparatus that we use to examine the slice. So here is the cell chamber, which contains the brain slice. Here's the microscope. This is the glass pipette that we use to connect to one of the cells. And then this is the Y tube, which administers chemicals to the cell chamber. So we'd examine the brain slice under the microscope. And then to connect to one of the cells, we would touch the cell with the pipette and apply brisk and brief suction to form a seal. On this graph, which is a graph of the membrane potential of the cell, you can see these lines, which is the membrane resistance, increasing, which means that we formed a seal. So then we apply some more suction to break a small piece of the membrane off and actually connect to the interior of the cell. You can see from this sharp drop in the voltage that we've connected to the inside of the cell because the interior of a cell is very negative. And then these spikes, this is what we would call firing, occurred because we administered current into the cell, causing it to fire. Okay, so here is a video of what a cell looks like when you give it an excitatory drug. Okay, so that tucking noise is what we hear every time the cell fires. And then, as you can see, the rate increases, meaning that it's been excited by a drug. Inhibition would cause the firing to slow down or completely stop, or even become more negative. Okay. So, on a graphing monitor, which records the membrane potential in real time, we would observe the changing voltage of the cell. And we would administer the chemicals to the cell through a Y tube, connected with one part inserted into the cell chamber and the other part, the bottom part, leading down into a waste bucket. Okay, so here's the graphing monitor and a lot of the equipment used to monitor the interior of the cell. This was very intimidating to me when I first walked in, but I eventually understood the uses for all these different buttons and cords and whatnot. So here is the graph of the GnRH cell's membrane potential when we administered MPY. As you can see, MPY had no effect on the cell because here's the time when we administered it for, and the firing continues to happen with no change in the rate or decrease in voltage. Here is the graph of the kisspeptin cell's membrane potential when we administered MPY. It's very evident that each time we administered MPY, the firing completely stopped, showing that it was inhibitory. And furthermore, we only administered it for about a quarter of the time as we did for GnRH. So this really shows us that MPY actually does not act on GnRH cells, but it actually acts on kisspeptin cells. Now we want to know which MPY receptors kisspeptin neurons have. To determine that, we tested several drugs that were either agonists or antagonists. An agonist is a chemical that mimics the response of another chemical. An antagonist blocks a specific receptor, preventing a chemical from acting on it. Agonists and antagonists have different degrees of selectivity, meaning it could act on all receptors or just one in particular. The first drug we tested is called LP-MPY, and it is a Y1 and Y5 receptor agonist. When we administered it, we saw that it inhibited the kisspeptin cell, as you can see from this graph here, which looks very similar to what happened when we administered MPY. So this tells us that either the Y1 or the Y5 receptor, or even both, are present on kisspeptin cells. But then we also tested a Y5 agonist, but this time it did not have an effect on the cell at all, telling us that Y5 actually is not present. So after testing several other drugs, we are able to deduce that MPY acts only on the Y1 receptor. Overall, we concluded from this study that MPY's effect on the reproductive system in the brain must be due to its inhibition of kisspeptin cells via the Y1 receptor. This can have several implications. For example, some patients who exhibit a certain MPY defect can overly inhibit kisspeptin cells, leading to infertility or delayed puberty, depending on the age. Now that we know that MPY acts on the Y1 receptor, we can potentially block its response by using a selective Y1 receptor antagonist. We would avoid interfering with the other properly functioning 4 receptors used for different purposes in the brain, thus reducing side effects. These implications have led me to more questions and possible future experiments I'd be interested to conduct. One of the topics that sparked my interest regards hypothalamic amenorrhea, which is when a hypothalamic problem such as hormonal imbalances, anorexia, overexercise, stress, or even medications causes acyclicity. I'm wondering, can we restore fertility to women with this condition using a Y1 receptor antagonist? Another topic I would inquire about is the cause of precocious puberty. Can we delay it? Precocious puberty, like I mentioned earlier, is an increasing trend in the US. Considering that MPY restrains puberty, could an MPY deficiency contribute to its premature onset? These are just a few examples of the many directions this study could take me. Lastly, I'd like to recognize all those who contributed to my experience at Yale. First of all, thank you. I guess I'd like to acknowledge my principal investigator, Dr. Meenakshi Alrija, Min Woo, who showed me the ropes of the lab, Dr. Gilmore, and Joanne Billiard for creating the program and investing so much time in each of the interns. And lastly, but not least, thank you, Ms. Monty. Your dynamic instruction, passion for science, and 100% support are what helped get me to this point. Thank you.